back, everyone, to the Phelan McDermott Syndrome podcast, sharing research, progress, and hope. For those of you I don't know, I'm Amanda Bergen. I'm the Director of Communications at the Foundation, and I have the honor and privilege to be able to record this specific episode while Dr. Lauren Schmidt is out on paternity leave. So really excited to be here with you guys today. And today we have three team members from Jaguar Gene Therapy, Dan Gallo, Kate Neer, and Gina Newton. We're so excited to have them here today to provide our Phelan McDermott Syndrome Foundation community updates on the JAG201 gene therapy trial. Thanks for coming on today.
Thank you for having us.
Happy to be here.
Awesome. Well, we want to jump into, individual introductions, but I think if we could just take a minute, I would love for one of you guys to share a brief, history and introduction of Jaguar as a company, but also a quick overview of JAG201. There might be some new families out there or some recently diagnosed families. And so really just taking a minute to pause and start there. What I would say is while JAG201 is one of several clinical trials specifically for Phelan McDermott syndrome right now, this is the first and only gene therapy trial available for individuals with Phelan McDermott syndrome.
Well, I can start, Amanda. I can give a brief overview of Jaguar as a company. So Jaguar Gene Therapy was founded in 2019 with the mission to accelerate gene therapies for patients living with severe genetic diseases. We're based in Lake Forest, Illinois, and that's about 30 miles north of Chicago. Our founding team was also the team that developed and brought to market the first gene therapy treatment for spinal muscular atrophy, or SMA. So we've got some experience there. After seeing the transformational impact that gene therapy had on families with SMA, our founders wanted to bring that same level of impact to other diseases. So that's why they created Jaguar. Currently, Jaguar has 3 gene therapy programs in our pipeline. First, of course, is JAG201 for SHANK3 haploinsufficiency, or Phelan McDermott syndrome. I'll let Dan provide more details on that program in a second. But we also have JAG101 for galactosemia. And galactosemia is a metabolic disorder that's caused by mutations in the GALT gene, which lead to a severe deficiency in functional GALT enzyme, and that causes a toxic buildup of galactose and its metabolites. This buildup of the toxic metabolites is a life-threatening medical emergency in newborns, and it can contribute to lifelong cognitive, neurological, speech complications, as well as primary ovarian insufficiency in girls and women. So JAG101 is currently in preclinical studies. We also have JAG301, which is for type 1 diabetes. JAG301, which is also currently in preclinical development, works to address the root cause of type 1 diabetes by producing functional beta cells using the PAX4 gene, which converts alpha cells to beta cells. So with that, maybe Dan, do you want to talk a little bit in more detail about JAG201?
Thanks, Kate. Happy to. JAG201 is a gene replacement therapy, and what we're aiming to do with JAG201 is to deliver a functional copy of the SHANK3 gene directly into the cells of the brain and the CNS of individuals with SHANK3 haploinsufficiency, or clinically what we call Phelan McDermott syndrome. A little bit of background, and some of this may be familiar, but SHANK3 is a protein that's expressed in neurons. It plays an important role in building and maintaining synapses, or the connections between neurons that allow communication. And then those connections are ultimately what make learning and processing and interpreting stimulus possible. So in PMS, individuals have only one functional copy of SHANK3 gene, again, what we call haploinsufficiency. And with only one functioning SHANK3 gene, there's not enough SHANK3 protein that's made from that one gene to support and maintain proper neuron-to-neuron communication and ultimately those connections between neurons. With JAG201, we're aiming to deliver, as I said, a functional SHANK3 gene, what we call a mini gene, directly into the neurons in order to increase the amount of SHANK3 protein that's available to build and maintain connections between neurons. There's multiple approaches to gene therapy. With JAG201, what we're doing is we're leveraging what's called an adeno-associated viral vector, often referred to as an AAV vector. And this is ultimately the vehicle or the vector that we use to deliver that functional SHANK3 gene into cells. And so just a little background on what that means. With a viral vector, it's called a viral vector because it's made up of certain but not all components of a virus. And specifically, it consists of the outside part of the virus, what we call a capsid shell, which is made of protein. And that shell houses the gene of interest, in this case, the SHANK3 mini gene. That capsid shell can interact with the cells of interest to deliver our gene. And then we use AAV for a couple of reasons. This is the one we've selected. It's effective at delivering genes of interest to target cells. And it's also largely thought to be non-pathogenic, meaning that even if in its natural form, it's not thought to be capable of causing illness.
Thank you so much for that. It's always good to rehear it, even as a more seasoned family member. But I really appreciate the overview and just all of the work that you guys are doing. It's really incredible. So thank you. So now that we're up to speed on Jaguar Gene Therapy as a company and the JAG 201 specifically, can each of you dive a little bit more into sort of your role at Jaguar Gene Therapy? And any background you think that you would like to share, but specifically, what part have you guys played in JAG 201 for Phelan-McDermid syndrome?
Sounds great. Yeah, I'm happy to kick us off. I lead our clinical development and our medical affairs groups at Jaguar. For background, clinical development is the part of the company that focuses on designing our trials and executing our trials. In this case, our clinical trial for JAG 201. And we work with the regulatory bodies like the FDA to gain agreement on the design of our trial with the goal of working towards an eventual approval for our treatment. The other part of the team that I lead is medical affairs, and that's the part of the company that's focused on communicating our science and our data to clinicians and researchers with the goal of educating and supporting ongoing research activities to address incoming requests for medical information. And eventually, if we can get to an approval, this would be the part of the group that would, again, educate clinicians to help them make informed treatment decisions with regards to how to use an approved treatment. In terms of my background, I've been with Jaguar for 4 years now. I'm a scientist by training. I hold a PhD in cell and molecular biology. Was at Northwestern for a large number of years working on that before moving into drug development. And I've been in drug development for about 15 years. Most of that time has been spent in the rare disease space. I've worked at both large pharmaceutical companies as well as startup biotech companies that are much earlier in development. Before coming to Jaguar, I led med affairs and advocacy for a rare disease focused company where I spent about 5 years working to bring a medicine to the clinic. It was the first FDA approved treatment for a rare pediatric neurodegenerative disease known as Niemann-Pick disease type C. I think I might just close my intro by saying, like many of my colleagues here at Jaguar, I'm completely inspired by our families in the PMS community. I think the goals and aspirations that families have for their loved ones affected by PMS are at the center of what drives me every day in my role. I truly believe we have a unique opportunity to make a difference in addressing the unmet need in PMS with JAG 201. And I feel privileged to be able to do this work and come to work every day with a dedicated team that is focused on doing everything we can to bring JAG 201 to the clinic as safely and as quickly as possible. So with that, I'll turn it over to Kate.
Jan. I lead corporate communications and patient advocacy for Jaguar. I joined the company in early 2021. I've spent the vast majority of my career in the pharmaceutical and biotech industry. I've worked with both rare diseases and large disease states, particularly with a long focus in brain disorders. In my role at Jaguar, I'm super grateful to partner so closely with the Phelan-McDermid syndrome community. In terms of the JAG 201 trial, I work to provide, I would say, the most up-to-date information about the trial to date. I am the one who works with the team here to issue our FAQ documents so that we can answer questions that were asked by community members. We also attend community events to be able to speak directly with parents and loved ones of those living with PMS. We attended the PMS Family Conference last year and are looking forward to attending again next year in Colorado. I also gather insights and feedback from community members on various aspects of the JAG201 program and make sure that those get shared with the broader Jaguar team so that they can be incorporated into our programs. For anyone looking to talk to me at any time, I can be reached at patientadvocacy@jaguargenetherapy.com. So definitely don't be shy. And like Dan said, the families whose loved ones have PMS are always at the forefront for me. I've learned so much in talking with individuals over the past, 4+ years about their families, journeys, their challenges, some of the triumphs, some loss and hopes. And so I've been incredibly privileged to be able to do that. And it definitely drives me every day. Outside of work, I'm a mom, although I guess also a mom during work hours as well. But I enjoy spending time with my family and occasionally, doing some cooking and baking and things like that. So anyway, it's great to meet those of you that I haven't met. And maybe Gina, do you want to introduce yourself?
Yes, thank you so much. So, hi everyone, I'm Gina Newton. I lead development operations at Jaguar. I've been with Jaguar for the past 2 years, and it's honestly flown by because this work is super inspiring, and it's just such an honor to work within the PMS community. I have spent about 25 years in drug development operations, between small and large companies. And I just, in general, feel so privileged to work on the JAG201 study. So development operations sort of sits within clinical development, and I really oversee the study to ensure that we're conducting the study with high quality and within regulations, and that we're collecting the highest and most complete quality data so that we can show evidence that JAG201 is safe and we hope effective. Like my colleagues, I can't stop thinking about the PMS families and communities. I am just in awe of all of you. I'm super impressed by the savviness of the families when it comes to the questions they ask about JAG201 and the trial. And it truly, this does not feel like work for me because of just what a wonderful community it is to support. So thank you again for having us on.
Thank you so much. It's so nice to be able to hear a little bit more about the work you're doing, but also just to, for those actually tuning in and watching this recording on video, be able to put a face to the name and to the work and to the, JAG201 that everyone's sort of so focused on. So I really appreciate that. And speaking of that, it leads me to the next question, which is, we've been meeting as PMSF and Jaguar monthly and regularly for some time now. And, as PMSF, our partnerships with industry and pharmaceutical companies like Jaguar are really focused on making sure that we are including the patient and the family voices and, making sure that that's shared back with you guys every step of the way. Can you take a little bit of time to share with our community the importance of the relationship with PMSF and some of the things that we've, consulted on?
Sure, I'd be happy to. I mean, first I'll just say Jaguar is very grateful to have built a strong relationship with PMSF over the past, 5 years now, I think. It's certainly not an exaggeration to say, and we say it all the time at Jaguar, that the JAG201 program wouldn't be where we are today in clinical trials without the help of the entire PMS community. A few, I would say, critical examples of collaboration that come to mind are the phase 1 clinical trial design. You guys at PMS provided feedback and reviewed trial synopsis and other materials before they were finalized. So you certainly brought the voice of the patients and families to that. You guys have also weighed in on logistics and travel that would be required for this phase 1 clinical trial that we're currently in to ensure, we're thinking about that family perspective. You guys have also served as I would say like a platform for us to reach the community so that we can share news and information about the JAG201 program. advising us on the best channels like webinars or, this podcast to be able to communicate directly with the community in the best way. Another example is how we've partnered and will continue to partner around natural history data collection and, different kinds of, information that we can get from families that will help us in the evaluation phase of the clinical trials with FDA regulatory authorities. So that will be hugely critical. I would just say you guys have really been there with us every step of the way, and we're super appreciative. So thank you guys.
Thank you. I mean, I think the feeling is mutual. We really are honored and privileged to have the relationship that we do. And I think I just want to take a moment and also thank our families because, without their voice and taking the time to ask the questions and share sort of what's on their mind and provide feedback along the way, because we know that, we wouldn't be where we're at today without that. So, also just thanking the families for that and for their time. So let's get more into the details on JAG 201. I think definitely things on people's mind and things that we've been hearing. But currently, we know the trial is open. It's at 3 sites, which we've shared with the community. So we have your first site that started at Mount Sinai in New York. We have Rush University in Chicago, and then most recently, Boston Children's in Boston. How do you guys think about site selection, and how did you choose these 3 sites?
I can address that. So, all 3 of these hospitals are obviously well-known, respected research hospitals, and Doctors Kovlevson, Dr. Barry Kravis, and Dr. Sirvastava care for many PMS patients and their families. And to be honest, I have never witnessed, research staff and physicians that are so committed and, dedicated to their patients and their families. All 3 of these physicians have weighed into the protocol, and, as we evolve through the trial, we learn things and we amend the protocol, and, we really sort of rely on them for their expertise. And so ultimately, these 3 physicians are just highly respected, super savvy with PMS, committed to their families, and they just really are on a mission as we are to bring these therapies to the PMS community. And again, we hope to determine that JAG 201 is safe and effective.
Thank you for that. And yes, I think you're right. A lot of people know those names and they are very well respected in our community. And one of the reasons why our community might know them is through the PMS natural history sites, so these 3 sites, as we know, are a part of that as well. Just taking a step backwards, I know we talked a little bit about this on our recent natural history study webinar, which we can drop in the show notes for those of you that missed that. But how do individuals' participation in natural history study help inform eligibility and other aspects of the JAG 201 trial?
I'm happy to speak to that a little bit and, I think give a little bit of background and also recognition, just how critically important natural history is and recognizing, those contributions and how much effort goes into generating natural history data. Much of what we know about the phenotype or the clinical presentation of PMS has been informed by data collected from the ongoing natural history study run by the DSC or the Developmental Synaptopathies Consortium. These learnings have been, for us, essential in helping design our first in human clinical trial for JAG 201. Whether it's the outcomes that we collect, the patients that we enroll, the duration of our trial, and even as Gina was just speaking to, our trial sites, all of this was in some part informed by learnings from the DSC natural history study. Without the natural history study, we really would not have been able to design our trial in the way that we have. And so in terms of why it's important, in rare disease drug development, we're often faced with a number of challenges. In particular, having to run trials in populations that are small, while at the same time, the large unmet need, the lack of approved treatments really highlight the urgency and need to move efficiently through drug development. Oftentimes, for ethical reasons or even logistical challenges, the standard type of trial you would run, which might be a placebo-controlled type trial design, may be infeasible in gene therapy and in rare disease drug development. In this case, natural history and the data collected in these types of studies provide potentially numerous pathways to accelerating drug development that may help overcome the need for, some of those challenges like a placebo-controlled trial. In our case, we are, as you mentioned, and we spoke about this on the recent webinar, we are supporting additional natural history data collection in slightly younger patients, so specifically age 12 to 36 months. And we're doing this at Mount Sinai, at Rush, at Boston Children's for a few different reasons. First, this is really a population where we're lacking data in the DSC natural history. So DSC, I believe they at one point open enrollment to patients as young as 18 months, but there's not a whole lot of data in the age range of, let's say, 18 to 36 months, and there's really none in the 12 to 18 month age range. And so collecting data in these participants will give us a better understanding of what development looks like in individuals with PMS at these earlier parts of their development. Generally speaking, these data will help us put into perspective any benefits or changes in development that we might observe in patients of this age that are treated with JAG201. So we'll have something to compare to, essentially untreated participants in the natural history. The second is, since these individuals are potentially younger than the ages that we might be enrolling in our first in human study, we may learn through the natural history that some of them may also be eligible for our study now or future trials. And in that case, their natural history and the assessments that they collect over the next months and potentially years would allow us not only to generate a comparison data set for our clinical study, but if they receive treatment with JAG201, it would allow us to go back and understand how did their development look in the months and years leading up to treatment, and how or did that change after they received treatment. Those types of data would be very powerful in helping to illustrate to regulators the potential benefits of JAG201. Our goal there would be to show that we're changing the trajectory of development within individuals treated with JAG201. So, for a lot of reasons, whether historical or the need to collect more data, natural history has been incredibly important from a drug development perspective and will continue to be important on our journey to bring JAG201 to the clinic.
Thank you. That's super helpful. And we often get a lot of questions on natural history. So just as a reminder, I guess, to share with everyone, we currently have the downward extension that Dan was just talking about, where there is enrollment available from 12 to 36 months. So we'll include all that information in the show notes. We'll include the webinar if you'd like to learn more about the natural history study. And then we're really waiting on a decision to be made on the possibility of opening it back up for a fourth round. And so stay tuned for anybody that's interested beyond that specific downward extension or 12 to 36 month age range for future possibilities. All right, so kind of shifting back to JAG201, we know that it's administered via ICV. Can you tell families a little bit about what that means and briefly about the procedure? And then kind of going back to site selection, process and phase, did you consider what experience each site had with the administration procedure?
Yeah, happy to. So yes, as you mentioned, JAG201 is administered via ICV or intracerebroventricular injection. And so if we break that down a little bit, ICV injection administration is an injection directly into the brain using a catheter or tubing to move that catheter into a space that's known as the lateral ventricle. And the way to think of a ventricle is it's a space, it's a cavity that sits within the brain that contains cerebrospinal fluid. And cerebrospinal fluid cushions and supports the entire brain and central nervous system. And so what we're aiming to do then is deliver JAG201 into that space so that it could be mixed within the CSF and then distributed broadly to all parts of the brain and CNS, which is where we want JAG201 to be able to interact with cells to deliver SHANK3. The procedure is performed by a neurosurgeon, so it is a neurosurgical procedure. It's done in the operating room under anesthesia. In our experience with our first patients treated, the procedure takes approximately 30 minutes. A neurosurgeon who's experienced at performing this type of surgery performs the procedure and has an understanding of how to guide that catheter into the right space, into the ventricle, By utilizing images that are taken of the brain using modalities such as MRI or CT. And so that's done beforehand so that when we get to the day of the surgery, the physician knows exactly where he needs to guide that catheter to get the most direct route into the ventricle and ensure that we're delivering the medicine to the right space. After administration, the patient's kept in the hospital for observation to ensure there's no unexpected outcomes of the procedure, and patients are generally discharged within about 48 hours. We selected ICV administration with the goal of maximizing how much JAG201 we could get delivered to cells of the brain, cells of the central nervous system. We want to reach as many cells as possible, understanding that we won't reach every cell. And so ultimately, this approach was selected, and it was informed by our non-clinical studies where we did experiments delivering JAG201 to the brains of different animals, utilizing the ICV procedure. And so, ultimately in those studies demonstrate that yes, we can reach the cells that we want to, we can do it safely, and then we could get to outcomes that we want to in the animals. And so that helped guide our decision to do it the same way in people. Um, as background, ICV administration is it's an established procedure. It's utilized in a lot of different disease spaces. It's utilized for short-term as well as long-term delivery of medicines when you want to increase the concentration of a medicine around the brain or in the central nervous system. And we've done a little bit of research just to get a sense of how common the procedure is, and we've seen publications that suggest that as many as 20,000 procedures, potentially more, are done annually in the U.S. And then, to the last part of your question, again, to go back to what Gina said, we try to be as thoughtful as possible in selecting our clinical trial sites. And absolutely, when it comes to the ICV part of the administration, we absolutely consider the capabilities of these institutions as well as their experience and the expertise within neurosurgery. So even though we're familiar with our clinicians who are investigators in the study, at every institution, that might not be the same individual that's delivering the medicine, right? It may be somebody within neurosurgery specifically. And so our team works closely with the neurosurgeon, with the clinical team at the hospital ahead of time in the weeks and months leading up to the trial to make sure that everybody's fully prepared for the procedure, and everybody's ready for, that day of administration.
Thank you. That's super helpful. And I think it's just really important to look at all of the different pieces that go into the entire trial, the site selection, and specifically who's administering what. So I appreciate you sharing that. Knowing that this current trial is a phase 1/2 trial, I did want to take a step back and just look at the clinical trial process as a whole, to make sure our community is all on the same page. Could you first remind families what a phase 1/2 trial is and what that means for this trial specifically?
Sure, I can take that one. So we talk a lot about phase 1 first in human, and that is because it is in fact the first time JAG201 has been delivered to a human being. So typically in a phase 1 trial, we are testing a small group of individuals for the very first time. And the goal of a phase 1 study is really to determine and learn about safety, tolerability, making sure there aren't any severe side effects, and then determining the ideal dose. So essentially, before this trial, any data collected on JAG201 was collected in animals. We call that preclinical data. And for JAG201, that was mice and non-human primates. And so ultimately, what we want to do in this phase 1 study is to determine the safe dose. So based on the preclinical data, we are looking at 2 different doses in the phase 1 study. And basically, this is going to help us understand the overall safety profile of JAG201, and then also determine that it's safe and we hope effective, and just really better understand, any side effects that a participant may have once they've been exposed to JAG201. And it's referred to as a phase 1/2 study because once we've determined that we have a safe dose, then we can potentially expand the study to treat additional participants at that safe and potentially effective dose.
Thank you. Which, you know, leads me right into sort of my next question, and you started to touch on it a little bit. So obviously, a huge component, like you said, is establishing that safety at the forefront of the trial, right? Like that's number one. But now that you guys have, began the process and we are underway with JAG 201, how are you beginning to think about efficacy? Or, like you said, in other words, how are we assessing whether it's quote unquote working?
Yeah, I can take that. So again, to go back to natural history, in designing our trial, we had the benefit of being able to leverage all of the learnings from the DSC natural history study. And so this includes the efficacy outcomes that we're evaluating. While we want to improve the overall developmental trajectory of individuals with PMS, we also recognize that this may take some time. And so, in evaluating the different types of outcomes that we wanted to look at, we set out to look for potentially earlier evidence of biological change. And then over the long term, also want to ask the question about, are we changing that developmental trajectory? So we understand from the DSC natural history data that EEG and specific measures of EEG, which looks at electrical activity and responses in the brain, may be abnormal in individuals with PMS. And so, one of the things that we thought early on was if you want to evaluate whether a treatment is changing that abnormal biology within neurons, one of the things that you may be able to evaluate is whether electrical communication within the brain changes after administration of a treatment, in our case, JAG 201. Again, we wanted to include this type of measure because unlike developmental milestones, which may take time to learn and to develop, we thought EEG may provide an early understanding that something in the biology of the brain is changing for the better. On the other hand, we know that FDA has a certain threshold that they look for when they're looking to evaluate whether a treatment and the data established from clinical trials justifies an approval. And so, when it comes to how FDA approves treatment, we know that they do so based on their ability to provide clinical benefit. And the way that the FDA thinks about clinical benefit is, does a treatment improve how somebody lives, functions, or survives? So, clinical outcomes that measure development, such as cognitive tests, communication measures, assessments of adaptive behavior, are all critical outcomes in our trial. These are also things that were collected in the DSC natural history. We know, how the population evaluated in the DSC natural history progresses over time on these instruments. And so in our trial, we're also looking at these types of measures, and we want to understand if JAG 201 treatment can help individuals reach developmental milestones and acquire new skills and abilities that they otherwise would not have been able to.
Yeah, thank you for that. I want to take a minute to pause here and also just really reiterate to our community and folks tuning in that the primary goal of a phase 1/2 trial isn't necessarily to reverse or cure the condition in such a small cohort of patients. The primary goal, as we sort of talked about, is that we need to establish that JAG 201 is safe and that there's target engagement, or in other words, as you discuss, it's getting into the brain, and that there's initial signs that it can change behavior and or development. I don't know if there's anything else to add there or anything you guys want to underscore.
No, I mean, I think you said it best. We really are focused on safety. I think all of us on the team, as I'm sure the community is, eager to know if it's working, but we're just not at that stage yet. So we're looking at, various assessments to make sure there aren't any significant side effects, that there's no worsening, from their baseline assessments. And, as Dan mentioned, we're looking at these validated scales, the Vineland, the SAND, which I know, are very comprehensive and, time consuming for the participants and their families. But, those are the scales that are really going to show us if there's efficacy. But for now, we really are focused on, safety, no changes in blood work or things like that. So again, like while we are all sort of waiting with you to see if this is, there's efficacy there, it's not our focus today. But, our goal is that once we determine a safe dose, then we would conduct additional cohorts to show if JAG 201 is truly effective.
Yeah. It's been such an interesting learning, even for myself, as a parent, to know every stage really does have its intention and it all builds up and works together. And, I think as families, we get really focused on sort of the end goal, but you can't get there without the different stages and the processes. So it's really helpful to sort of be able to walk through that and talk, a little bit more about it in depth. Um, so part of the process of designing a phase 1/2 trial is selecting inclusion and exclusion criteria. Probably one of the most common questions that we've heard from families. I think you guys as well have heard a lot of these questions too. So just for those tuning in today and for our community out there, walk us through the decisions that you guys make on the current inclusion exclusion criteria. Specifically, the young age range is one that we hear a lot about. And then this class 1, class 2 deletion, which I think is newer language in a clinical trial for our community. And we've done some education pieces around that as well. But how did you guys think about those things when sort of nailing it down for JAG 201?
Yeah, I'm happy to speak to that, and we could break it down and make sure we touch on each of those. Um, I think I'll start by saying our intent with JAG 201 is to develop a treatment for all individuals affected by PMS. But we have to start at a particular place. And so, that's, I think how to put into context what we might see in the inclusion and exclusion criteria. In designing our first in human trial, we recognize we have this one first opportunity to demonstrate whether or not our treatment is safe, but then also to explore the potential for benefit in this population. And so success in a first in human trial then opens the door to further evaluation in larger populations, which would result potentially in changing criteria. And it also gets us closer to the goal of an approved treatment. So just to highlight, we want to be as thoughtful as possible in determining the characteristics for those first few participants. And again, this is really just a starting place. So let's break it down. There's a few different reasons for why we select these different criteria. So starting with age, we recognize from the natural history that there's perhaps increased ability to acquire developmental milestones and skills in individuals with PMS earlier in life. And so while milestones may be reached later, individuals we know still have the potential to acquire milestones. So we know that there is also a potential to lose certain skills and abilities via regression. Biologically, we don't fully understand why. So in selecting the age, we really wanted to find this ideal spot potentially where we could enroll participants that maybe have greater potential for benefit just due to the fact that they're actively undergoing neurodevelopment, but then also be prior to any potential onset of regression. And so the goal here then is let's demonstrate it's safe. If we can have some success here in a younger population and there's some signs that JAG 201 is doing something that benefits patients, this would then give us the confidence to evaluate that benefit in potentially an older population. Um, and then we'd have the opportunity to then compare between those two, right? Do we see similar levels of benefit in younger patients as we do with older? On the other hand, if we were to start, in somebody much older, I think we might have question of, well, is the potential for benefit there the same as somebody who's much younger? And if we don't see a benefit, does that give us confidence to move in the other direction? So we really want to start where we think there's a potential to show benefit, where we, have confidence in the safety, and then we can expand from there. As far as genetics, with JAG 201, we know that we are only providing a SHANK3 mini gene. And so in our first study, we wanted to focus on patients whose genetics reflected primarily the loss of a SHANK3 gene or a SHANK3 allele. Again, demonstrating benefit here would give us confidence in the ability of JAG 201 to address SHANK3 haploinsufficiency, and then that would pave the way to evaluating benefit in individuals with, let's say, larger deletions. As you know, deletions can be highly variable, right? Everyone's deletion is going to be different potentially. So with larger deletions, there are additional genes that are impacted. Some of those genes may have an impact on development. And so in our first study, our first few patients that we treat, we want to avoid a situation where the genetic background of the individual may make an assessment of benefit uninterpretable. So is it that, the gene worked, but there were too many, things impacted in this very large deletion that now we can't see the benefit? We wanted to avoid that type of situation. Lastly, and I think this one's come up more recently, is with regards to other investigational treatments. In general, when designing these types of studies and earlier trials, especially, sponsors exclude participants who've recently received or are receiving potentially disease-modifying investigational treatments. Exclude them from clinical trials when you're evaluating new potential treatments. And the reason for this is that doing so reduces the possibility that confounding factors may impair the ability to interpret the outcomes. In other words, excluding exposure to other investigational treatments will help give the regulatory authorities the confidence that any benefit that we see in our trial, or, on the other hand, any adverse effect that we see in our trial is being caused by the therapy that we're investigating. So in our case, JAG201, and not another medicine where, the clinical profile or exposure to that medicine and the results of that are not fully established. So to that end, we've received to that end, those who've received treatment or are receiving treatment with potentially disease-modifying treatments would not be able to participate in JAG201 clinical trials. And this does include things that are currently in development, including Neuren's NNZ compound. Uh, Again, the approach here gives investigational treatments the opportunity to be independently and then appropriately reviewed by regulatory authorities. And it's important. What we want to do is get to an approval as quickly and as safely as we can. And we want to avoid at all costs a situation where FDA is evaluating our clinical data, but then can't be sure of how to interpret it and whether the benefit or any negative impact is due to our drug or another. What would generally result in that type of situation could include even FDA asking us to go back and do additional studies in a specific population that eliminates those potential confounders, again, to justify a potential approval. So, the more rapidly and safely we can potentially get JAG201 approved, our hope is, then the sooner that all families can consider it as a treatment option, once we get to that point.
Yeah, I appreciate that. And I think it's such a good reminder for families about sort of the intention behind, the clinical trials. But just because the inclusion exclusion criteria today for phase 1, 2 is such, of a certain age group or specific, deletion size, it doesn't mean that in the future that there might not be the potential to be able to have this therapy should it go, to market. So I appreciate that. All right. So I know that, we've been keeping our families waiting. And I know there's still a lot that you guys can't share, but any progress updates that you can share with our community?
Yeah, I'm happy to give a little bit of an update. And some of this, maybe families are familiar with based on, some of the announcements that have come through PMSF. But I'm happy to share a little perspective on where we are. So to date, we've dosed our first two patients. From a safety perspective, we've reported that there have been no serious treatment-related adverse events. Both of our patients continue to do well, and we're optimistic about the potential for benefit. Patient one, participant one, is about 6 months out from their dosing date. And then our second dose participant is closer to that 3-month mark post-treatment. We are currently on track to dose our third patient in the next month or so, and we're aiming to complete enrollment in this first-in-human trial by about the third quarter of next year. We are really looking forward to sharing more updates on the outcomes, including some of the stuff that we talked about here today. And ultimately, we'll be planning to present preliminary data from our patients at conferences in 2026.
Yeah, we're excited to have you guys there. And what an amazing milestone. Thanks for sharing that update. And we look forward to hearing more as it continues to evolve and you guys get more information. Um, where are we at for next steps? What does this mean for next steps? What can we expect for the remainder of 2025, heading into 2026? Any details there that are open to share with our community?
Yeah, again, priority will be to continue to follow our patients, make sure that everybody continues to progress through the trial safely, and then continue to collect data over the coming months and years. ultimately, as I said, we aim to complete dosing for this first in human trial in about the third quarter of next year. And then with the data that we collect in these patients, we would aim to engage with regulatory authorities, the FDA, as early as next year to start to gain alignment on the pathway to a registrational trial. And that trial would ultimately generate the data that would support an approval. So I think as we get a little bit further along in our dosing cohorts and have an opportunity to evaluate the efficacy and safety outcomes, we'll definitely be looking forward to share additional updates on the path ahead with the community.
Yeah. And to dive a little bit more into that, so I think that's really great, and we will continue to share that information out. As you think about this next phase and the potential of where it could go, we talked about this a little bit ago, but I guess I'm going to double click here on the inclusion criteria. Could it potentially expand for the next phase? Also, another question that we get quite a bit is, what about expansion to other countries? Are you guys thinking about any of those things as you sort of build out the pipeline and think about sort of the future?
Yeah. So yeah, both great questions, and of course we're always thinking about these. We know that these are top of mind for families. As we mentioned, our goal here is to ultimately utilize the data that we generate in this first trial to gain agreement with regulators on the design of our registrational phase trial, so the trial that would support an approval. Within that registrational phase, our goal, we'd also be aiming to expand the inclusion criteria to potentially include some of the groups that we mentioned, potentially individuals with larger deletions, individuals older than 2 to 9 years old. We're definitely looking forward to being able to share more about our current trial, including outcomes from the first patients that we've treated in the months ahead. And again, depending on these outcomes, evaluating how we can expand that population, really underlining that in developing JAG201, our goal remains to develop a treatment for all patients with PMS. We don't currently have plans to expand our trial into other countries, but certainly we're thinking about how we get to an approval and what a pathway looks like for an approval here through engagement with the FDA. And should we find a path to approval, that would open up the door to approvals and access potentially in other countries.
Thank you.
To Dan, if I could add, sorry, just to, as we're thinking about the future and what that looks like, with all this work ahead of us, right? As Dan just outlined, it's going to be super important for us to maintain the close relationship we have with you guys at PMSF and the whole community. So, we're going to have to continue to rely on your feedback and insights and experiences, because that will inform the JAG201 program as it progresses over the next months and years. And we'll also continue to be transparent and timely and consistent in informing everyone of where JAG201 is with its progress. As Dan said, hopefully we can share additional data next year with the community and the public in general. And as a reminder, I just wanted to say too, we've mentioned it a few times that you can access our latest FAQ documents with, via you guys with PMS. Certainly you guys always have our latest information. We also have it on our Jaguar website on the patients and caregivers page. So you guys can always check there. We do try and update it, with the latest questions. In fact, we as a team, I know, Amanda, we talk a lot about, does it make sense to add a question in or update a question's answer based on questions that we're getting, both you guys and we're getting from the community. So we try and do that often as well. So like I said, the latest version PMSF always has, and then you can find it on our website. But, again, this partnership for us is very, very critical. And I just wanted to say thank you for that now, but also for that in the months and years to come, because it's going to continue to be super critical for what we're going to be doing.
Thank you, Kate. I really do appreciate that. And I mean, even this morning as we had a meeting together, as our regular monthly meeting, it's always so great to be able to have those conversations as we think about the future. And our mission at the foundation is bringing those family voices and patient stories forward and really considering them every step of the way. And so, as we went through today, I hope the message is clear that, safety is always number one. but we're not forgetting about all the other aspects of the daily lives that our families, deal with day in and day out. And, as much as we continue to hear from this community that we're speaking with today, we will continue to bring that to Jaguar. And I know how much you guys take it to heart. So we appreciate that partnership and that continued conversation and collaboration. So it's definitely mutual. Going back a little bit to the timeline, I just want to sort of wrap up that conversation because I do know that this is an area where you hear 2026 and 2027, and in some ways it feels so exciting, but it also can feel a little discouraging to our families in knowing how long that this process can be. Science is never going to be fast enough, right, for those that live with Phelan-McDermid syndrome day in and day out. I know I can speak to that as a mom and caregiver of someone with Phelan-McDermid syndrome. But I also want to highlight the fact that this is the first in-human gene therapy trial that has started for Phelan-McDermid syndrome. It's such a huge milestone, and nothing can get to the market without these initial steps that we discussed today. And so it is so important to talk about what that looks like and why we have to go through all of these things. And I was reflecting back on my journey. My daughter was diagnosed in 2017, and so it's been almost 8 years. And when she was diagnosed, there was nothing on the horizon. I remember feeling a little bit hopeless, and just to see even where we've come in these 8 years, it really is incredible, and it's really an exciting time. And so we appreciate, again, all the work that you guys are doing and the partnership that we have, because we wouldn't be here today without it. So I just want to take a minute and say thank you so much, Dan and Kate and Gina, for being here today. I know that we will continue to eagerly be waiting more and on the edge of our seats for more updates. And we'll continue to communicate with and to our PMSF community anything that comes our way on recruitment needs to our community, as well as updates. So thanks again for being here. Thanks to the community for tuning in. And until next time.
Thanks so much, Amanda.
Thank you.